Hello dear students, welcome back to biology class. Chapter 6, Molecular Basis of Inheritance. In the last video, we discussed about Griffith experiment and Oswald Avery, MacLeod and McCarthy experiment. In this video, we are going to discuss about Hershey and Chase experiment. Hershey and Chase experiment. There were two scientists, Alfred Hershey and Martha Chase, in 1952 conducted and blender experiment to prove DNA is the genetic material. For their experiment, they used two organisms. One is bacteriophage and other is E. coli bacteria. This is the bacteriophage and this is the E. coli bacteria. The bacteriophage means the virus that infects the bacteria are called bacteriophage. Structurally, the bacteriophage having two parts. One is the genetic material and other is the coat which surrounds the genetic material called protein coat. It has mainly two parts. One is genetic material, that is DNA, and other is protein coat. As I have told, the bacteriophages which infects the bacteria. When it infects the bacteria, first it attaches to the bacterial cell wall. After that, it inserts its genetic material inside the bacterial cells. The Hershey and Chase conducted the experiment to know whether the DNA of the bacteriophage enters inside the bacterial cell or the protein coat of the bacteriophage enters inside the bacterial cell. Next, about the experiment. They conducted experiment in two setups. In the first setup, they grew some bacteriophages on radioactive phosphorus media, where the DNA of bacteriophages become radioactive because phosphorus is present in DNA. As already you know that phosphate group is one of the component of nucleotides. Hence, the DNA is become radioactive. In this diagram, the red coloration indicates radioactive DNA. In the other set, they grew some bacteriophages on a radioactive sulfur media, where the protein coat of bacteriophages become radioactive because sulfur is present in proteins. Hence, the protein coat is become radioactive. This red coloration indicates radioactive protein coat. In the next step, the E. coli bacteria are added into the media. The radioactive phages, that is radioactive DNA containing bacteriophage and radioactive protein coat containing bacteriophages, are allowed to infect the E. coli bacteria. After the infection, the phages transfer their genetic material into the bacteria. The question is whether the DNA inserts inside the E. coli bacteria or the protein coat. In the next step, the media is agitated in a blender machine because the bacteriophages are attached with the bacterial cell wall. It is necessary to separate the bacteriophages from the bacterial cell. That's why the media is agitated in a blender machine. In the next step, the culture media is centrifugated in a centrifuge machine to separate the bacteria from viral particles. In the centrifugation machine, high density particles or heavy molecules are deposited at the base called pellets or deposition. In the upper position, the low density molecules or light particles they used to float. In this experiment, the bacteria having the higher density, the bacteria used to deposit at the base. The bacteriophages having the lower density, they usually found in supernatant. Observation. In the first setup, the radioactive DNA containing bacteriophages allowed to infect the E. coli bacteria, where after centrifugation the deposit is radioactive. The supernatant is not radioactive. As already I have told, in centrifugation process, heavy molecules or high density molecules deposit at the base, low density particles are float at the upper region, where the bacteria having the higher density, they deposit in the base. The viruses having the lower density, they used to float in the supernatant. In the first setup, the deposit or the pellet is the radioactive. That means bacteria are radioactive. In the second setup, the bacteriophages having the radioactive protein allowed to infect the E. coli bacteria, where the radioactivity is seen in the supernatant but not in the deposit, because in the second setup protein coat is radioactive. Hence, it means that protein coats are floating in the supernatant. They does not enters inside the bacteria. Hence, the basal bacteria are not radioactive. In first setup, the deposit or the pellet is radioactive because DNA is inserted inside the bacterial cells and bacteria are become radioactive. In the second setup, deposit is not radioactive because in second setup, protein coat is radioactive and it is floating in the upper side. Hence, from this they concluded that DNA is the infecting agent that made the bacteria radioactive. Hence, DNA is only the genetic material but not the protein. Let's watch an animated video of Hershey and Chase experiment. Hershey and Chase experiment. For their experiment, they used two organisms, bacteriophage and E. coli bacteria. Bacteriophage is the virus which attacks E. coli organism. There were two scientists, Alfred Hershey, Martha Chase, conducted experiment to prove DNA as a genetic material. In their experiment, they grew some bacteriophages in radioactive phosphorus media, where the DNA of bacteriophages become radioactive. In the other setup, they grew some bacteriophages in radioactive sulfur containing media, where the protein coat of bacteriophages become radioactive. In next step, they allowed the bacteriophages to infect the E. coli cells. After infection, they insert their genetic material into the bacterial cell. After that, bacteriophages are separated from bacterial cell wall by means of blending in a blender machine. In next step, the radioactivity is detected by means of centrifugation. In the first setup, where the bacterial cells are become radioactive because the radioactive DNA are inserted inside the bacterial cells, whereas in the other setup the bacterial cells are not radioactive because in the second setup protein coat is radioactive. Protein does not enter inside the bacterial cells, hence bacteria are not radioactive. In the first setup, bacteria are radioactive. From this, they concluded that DNA is only the genetic material. Thank you for watching the video.